Hello! Our iMeta review paper is entitled Applying Multi-omics Towards Tumor Microbiome Research. The tumor microbiome is an emerging topic in cancer research, with research aiming to better understand how microbes, including bacteria, archaea, and fungi, may contribute to carcinogenesis, cancer development, and metastasis, and may even modify cancer therapies. Recent papers have shown that these microbes can affect cancer through many methods, some of which are shown here. While the tumor microbiome has shown great promise as a target for the early diagnosis and treatment of various cancers, there are still many limitations, including the low biomass of intratumoral microbes and the unculturable characteristic of some species. For this reason, new methods that can overcome these challenges with higher temporal and spatial resolution are urgently needed. In this paper, we discuss how various omics techniques like genomics, transcriptomics, proteomics, metabolomics, and a combination of these techniques through multi-omics could make an impact on the field. We begin with the omics methods aimed at the earlier levels of the central dogma, which are genomics and transcriptomics. These methods focus on nucleic acids, mainly those that make up DNA and RNA. Currently, next-generation sequencing and third-generation sequencing methods are utilized for a variety of these techniques, including DNA sequencing, RNA sequencing, 16S rRNA sequencing, epigenetic techniques including ChIP-seq and DNA and RNA methylation sequencing, and three-dimensional genome sequencing. Additionally, recent advances in genome editing have become important tools to study the tumor microbiome and tumor microenvironment, thereby improving the identification and traceability of these tumor microbes. Single-cell techniques aim to reveal gene structures and gene expression of a single cell, better reflecting the heterogeneity between different cells. In regards to the tumor microbiome, microbes of interest may be of a lower abundance, and thus may be covered by the signals of higher abundance organisms, which may lead to a loss of these signals during characterization. Single-cell methodologies, including agar dilution plating, single-cell exome sequencing, droplet-based microfluidics, and improved fluorescence in situ hybridization may serve to better study these low-abundance microbes. Moving forward in the central dogma, proteomics serves to study proteins and their characteristics such as expression levels, post-translational modifications, and interactions. Mass spectroscopy coupled with liquid chromatography, also known as LC-MS, has become a popular method for such analyses. The two broad approaches towards shotgun proteomics, data-independent acquisition, DIA, and data-dependent acquisition, DDA, can be both utilized for tumor microbiome research. DIA tends to be popular to study pathogenic organisms and clinical samples, particularly when looking at understudied organisms or complex tissue samples. DDA is more widely used and tends to be important for targeted analyses. Metabolomics can be used to quantitatively analyze many metabolites and to discover the corresponding pathophysiological functions of these metabolites. The most common metabolomics methods currently include nuclear magnetic resonance, NMR, LC-MS with multiple reaction monitoring, and gas chromatography mass spectroscopy, GCMS, with selective ion monitoring. NMR is a non-destructive method that has easier sample preparation, but is limited by its low sensitivity, the complexity of microbial metabolites, and the variation in metabolite concentrations. On the other hand, high-resolution mass spec has been able to overcome some of these problems, with GCMS being used for volatile and thermally stable low molecular weight compounds, and LC-MS being used for polar and high molecular weight compounds. Finally, single-cell proteomics and metabolomics are emerging methodologies, though these signals cannot be easily amplified. With the field being new, it still lacks a clear approach, though there has been rapid progress. We are growing more aware that a single field of omics is not comprehensive enough to understand more complicated biological processes, such as those related to the tumor microbiome. Integrated multi-omics seeks to fill this gap in research and is a potential new direction to explore these topics. Although many advances have been made in the field through omics approaches, this field is still in its infancy. Many research techniques are immature or still theoretical, though we propose that single-cell multi-omics will become the most powerful tool in tumor microbiome research, though there's still a long way to go in developing the corresponding techniques and methodologies.